I've given you a key term.
Respiration. Oh, that's next week.
Inheritance. You're doing respiration on Monday with Miss Wolf. Although Dr. Biology has done a good series of videos on respiration. Yeah. Right. So there we go. So there is a sheet with some. Sorry, the reason I'm filming it for other students. Okay. So I think key terms are really important in this section of work. Okay. Knowing what homozygous, heterozygous, monohybrid, dihybrid, haploid, diploid, and all that. So rather than kind of giving it all mixed up, I've given you the answers and the, you know, so you could cut them up if you wanted to test yourselves. That would be good. This crib sheet, this has all the different things you need to know in this section of work for inheritance. Okay, so it's quite confusing, all the things you need to know. So I have separated them out into all of the things and what they are. You would need to know, you would need to know about Punnett squares as well. And then also I've included a revision kind of tick list as well. Okay, so all the things you need to know. Good. Right. Obviously, in this time, I'm not going to be able to go through the whole of inheritance. So I'm going to go through some elements of inheritance that I think are important and come up regularly in questions. So let's have a look at what I'm going to do. Okay, so the first one I want to talk about is codominance. Okay, so I'm going to do a little mind map. It's up to you. Again, this is because it's voluntary. If you just want to listen, that's fine. If you want to write things down, that's fine. What I will ask is that you don't, you know, you can ask each other questions, but don't chat about other stuff. Okay, because we've only got a limited stuff. We've only got a limited time together. So codominance. So basically, that's saying that the alleles, okay, are equally dominant. Okay. If you have any questions as well, please do ask. So they're equally dominant and they're expressed in the genotype. Yeah, so genotype is actually the combination of alleles within a cell. The phenotype is the observable characteristics or biochemical characteristics of an organism. Okay. So I'm going to give you an example here. Snapdragons, I think, is a popular one. So, for example, we have FR. That's not good. FR is red. FW is white. Right. So how would we do a cross? How do you do a cross of those things? What would we use? Yeah. We do the Punnett Square. So I'm going to give you, I would like you to do a cross between a white individual with a red individual. Okay. So those are the genotypes of the parents. Okay. So I'd like you to work out, please. First of all, cross them. What's the proportion of phenotypes? So what will we get from that? Okay. Right. Okay. So what have we got? Okay, so they're all the same. Okay, so every one of them is 100% pink. Yeah, so they'd be the phenotype is pink in this case. It's 100%. They would be pink. Okay. Now, usually you then would cross these with each other. So I'd like you now to do the following cross of FRFW with FRFW, please, and work it out again. Give it a chance. Wait a minute. Okay, so we would write it out. So that would be red. That would be pink. That would be pink. And that would be white. Okay, so we can say it's basically 25% red. So it's a 1-to-1 ratio. So that's red, that's pink, that's white. Okay. Okay, I'll just give you an example as well of codominance. So things like sickle cell anemia, the sickle cell anemia. Now, do you need to know what sickle cell anemia is in your spec? No, you don't. Okay, so what I'm saying is that is used as an example, but they could use any example. All right, it will be really clear that it's about codominance though. So sickle cell anemia is when you have sickle cell traits where it affects the shape of red blood cells. So the red blood cells are sickle shaped, and that means that the hemoglobin is affected. Okay, and then you can get differential kind of differential sickle cell traits. And what they'd like to show you is some data, usually about Central Africa. So if you see pictures of Central of Africa, it's about malaria and it's about sickle cell, it's codominant. So multiple alleles. So this is when it gets a bit more interesting. So multiple alleles is when there's more than 2 alleles that may be present on an individual's chromosome. Okay, so more than 2 alleles. Traits of more than 2 alleles. Can anyone remember a good example? It's blood groups. So blood groups, you can get lots of different alleles. Okay, 3 different alleles. So 3 alleles affect antigens and red blood cells. So you can have IA and IB. They're codominant. And then you've got a recessive one as well. And then IO is recessive. Worth knowing that. Again, they're very applied questions. Right, let's do an example. I'm going to tell you a parent's genotype. I'd like you to tell me the phenotypes, please. So parent A is IO, blood group for the male. For the female, they are AB. So I'd like you to cross those, please, into Punnett Square. Thank you. You go.
Is the male IO and IO double IO?
So yeah, it would be. So what have you said the male is?
IO.
Yeah. IO. What would the female be? IA. IB. Well done. Okay. Okay, so you should have got the following. Doesn't matter which way round your alleles go. They can go the other way. It doesn't matter. It's quite an interesting mix there. So what do we notice? So 50% are A, group A, okay? 50% group B. There is no group O. Okay, so there's a 50% chance. So let's cross an A and a B. Okay, so we're just getting all we're doing is we're practicing being able to draw Punnett squares here. Okay. Okay, so we would have IA, IB. That's AB. We have A because it's dominant, B because it's dominant, and O. So you'd have AB, A, B, O, 25% each. Okay. Right, let's get on to my favorite thing that always comes up. I want to talk about sex linkage. When we talk about sex inheritance, we always talk about, well, what are the chromosomes for a male and a female? What's a male? XY. So XY is usually a male, usually, okay? But I'm going to tell you, and I think I shared with my my class at the time, read the question really carefully. There was a question that had chickens whether the male was XX. So if it says the male is XX, the male is XX, okay? So what I'm saying is go with the stem of the question. So be careful reading the question because it might not be what you expect. It was, I think it was about chickens actually. Just be careful, okay? So usually it's 50%. We know about that, sex inheritance. But we need to talk about sex linkages, okay? So a few things about the X and Y chromosomes. So X and Y. So what do you notice about the X chromosome compared to the Y? It's the Y is shorter, so X chromosome is longer. Okay, it's got more genes than Y. Okay. So that means there are many. Okay, so there so there's many homologous portions of Y missing. So they're not found on the Y, but they're found on the X. Okay, well, because you've only got half the chromosome, if there's a gene there, okay, there's a gene there, there's a gene there, there's a gene there, it's not there. Okay, it's missing. Okay, so that's how we get changes in terms of sex inheritance, because the homologous portions are missing. Okay, that's why you have less genes and therefore it affects the outcome. So Y controls very little. Right, so let's look at Drosophila. They love Drosophila. So what is Drosophila? Fruit fly, Drosophila melanogaster, fruit fly, and they like to look at eye colours. That's red eyes. That's white eyes. Right, I'd like you to cross those, please. Thank you. And we'll talk about the outcome.
So it's only the colour of the eyes is determined.
Right, okay. So let's have a look. So X, E, Y. So what's really strange about that? What's strange about that?
They're all white eyes.
They're not all white eyes. All red eyes. So all red eyes. 50% male, 50% female. They are all red eyes, okay? Okay, see you later. All right. Well,